Fission in amoeba. Amoeba uses the method of fission for reproduction. Here in the first is a mature amoeba. In the stage 2, here the nucleus elongates. Then in stage 3, a constriction appears in the cytoplasm here. This constriction then deepens, and as the constriction deepens and we get two daughter nuclei, two daughter cells are formed. This method is called binary fission because one cell divides into two. So binary fission can be defined as splitting of a single cell into two daughter cells. In amoeba, however, the division takes place along any plane. The next we will see about Leishmania. It also undergoes binary fission and it's a little different from amoeba. In this slide we'll learn about fission in Leishmania. Leishmania is also a unicellular organism. This Leishmania causes a disease called Kala Azar. Unlike the amoeba, Leishmania shows more organization. Like it has got a whip-like structure. It's got a definite structure, a definite shape, and this whip-like structure is always present at the end of the body. Now, Leishmania also undergoes fission, like you can see in this picture, but the fission takes place along a definite plane. And if you've seen here, it takes place along this plane. It will not take place along this plane, the horizontal. It always takes place along a definite plane. But here too it is called binary because one Leishmania breaks up into two. So the fission in Leishmania takes place along a definite orientation in relation to these structures. Unlike amoeba, the fission took place along any plane, but Leishmania it takes place along a definite plane. Here is fission in Plasmodium, and the fission in Plasmodium is called multiple fission because a single cell gives rise to a number of cells. Now Plasmodium is a malarial parasite. It's a parasite that causes malaria. Here is a picture of Plasmodium, and this Plasmodium is always surrounded by a cyst, a protective covering or the cyst. Now in this picture next we can see how a single Plasmodium, first within the Plasmodium you have a number of little cells that are formed. The nucleus undergoes multiple division and each of this nucleus gets surrounded by cytoplasm, and then later the cyst breaks open, releasing all the malarial parasites. So this division is called multiple fission because a single cell gives rise to a number of cells. Let me explain this point again. Here first we have the nucleus. The nucleus first gets divided into this many nuclei, and then the cytoplasm that surrounds this nucleus then slowly surrounds each of these nuclei, forming this number of cells, and later the cyst breaks open and releases these daughter cells. So in multiple fission, a single cell gives rise to many daughter cells, unlike binary fission in amoeba and Leishmania, where a single cell gives rise to just two daughter cells. Fragmentation. And from the word we know it is breaking up into smaller bits. Fragmentation is shown by Spirogyra. Here is a picture of a Spirogyra. It's a thin, long strips. And this Spirogyra, like you can see here in the next picture, this Spirogyra just breaks up into different segments. Here it is broken up into 3 fragments. And each of these fragments then grow up into an separate individual. A long separate individual, and this is called fragmentation. This method is shown in organisms which have simple body structures. So Spirogyra simply breaks up into smaller pieces of fragments upon maturation. Each of the fragments then grows into a separate individual. Here is yet another method of asexual reproduction called regeneration. From the word regeneration is to generate again. Like in the first case, we can see the planaria. When the planaria is now cut into 3 bits, and each of these bits then develop into a new planaria. The same thing, hydra. Hydra is cut into 3 bits, and each of these bits then develop into a new hydra. This is a picture from your textbook. So here you have planaria, which is cut into 3 bits, and each of these bits then develop the missing parts. Have you seen? Here the missing parts. Each bit develops some missing parts, giving rise to 3 planaria. Starfish also reproduces by the method of regeneration. Now this is a little note from your textbook. Now hydra, planaria can be cut into any number of pieces, and each piece grows into a complete organism. This is known as regeneration. Now regeneration is carried out by specialized cells, this is important, called the regenerative cells. These cells proliferate and make a large number of cells. From this mass of cells, different cells undergo changes to become various cell types and tissues. Another point you must know is that regeneration is not the same as reproduction. Why? Since most organisms would not normally depend on being cut up to be able to reproduce. Like when you have the planaria, it has its own method of reproduction. But if at all it is cut up, then each piece will develop into a new individual. The same thing for the starfish. The starfish has its own method of reproduction. But if the starfish is cut up, each piece then develops into a new starfish, and so also for hydra. So it cannot be considered as a method of reproduction. Now for another method called budding. It's from the word you can see here a bud is developed. It happens in hydra. In this location we have the regenerative cells. These regenerative cells divide and redivide, and they form this outgrowth called the bud. The bud then slowly develops into a new individual, and once it is mature, it will then cut out and you get a new hydra. So in bud formation, a bud develops as an outgrowth due to repeated cell division at one specific site. The buds develop into tiny individuals, and when mature, they detach from the parent plant and become a new individual. But what is important, they need these regenerative cells. The cells at this site will multiply repeatedly to give rise to an outgrowth called the bud. The bud then develops into a new individual here, and once the individual is mature, it cuts off from the parent and grows on its own. The next method is vegetative propagation. Before we learn vegetative propagation, we must know that the plant has got 2 parts, the vegetative parts and the non-vegetative parts. The vegetative parts are like the roots, stem and the leaves, and the non-vegetative parts are like the fruit, the flowers, which are involved in sexual reproduction. Many plants use these vegetative parts like the stem, roots and leaves for their reproduction. So that reproduction is called vegetative reproduction. So it's a method of asexual reproduction involving vegetative parts like the stem, roots and the leaves. Here is an example of vegetative propagation, like the cutting that we do so often in our homes. We take a stem of a plant, we cut the stem and we grow it into a pot, like in the case of roses, sugarcane, grapes. These all plants are reproduced or multiplied by vegetative propagation. So here are some examples of vegetative propagation by stem. Then this is another example of the bryophyllum. And here in the bryophyllum, you see these buds grow in the notches of the leaf. And they later develop into a new plant. So this is also a method of vegetative propagation. Here the bryophyllum. Then many other plants like the strawberry, you have the onion, they all grow or multiply by vegetative parts like the roots, stems and the leaves. The next question is, why do plants reproduce by this method? What are the advantages of vegetative propagation? I've taken this from your textbook. The first advantage of vegetative propagation, number one, is the plants that reproduce by vegetative propagation can bear flowers and fruits earlier than those produced by seeds. The second advantage is such methods also make possible the propagation of plants such as bananas, orange, rose and jasmine that have lost their capacity to produce seeds. Like the orange seed, if you grow the orange seed, it is not going to germinate. The rose, the jasmine, these seeds have lost their capacity to propagate. So these plants then use vegetative propagation for reproduction. Another important part of advantage of vegetative propagation is all plants produced are genetically similar enough to the parent plant to have all its characteristics. So for example, if we have an apple tree that produces juicy apples and we want to have an orchard of such apple trees with exact same characters, here we will use vegetative reproduction, not sexual reproduction, which we will learn later. We will use vegetative reproduction to produce another plant which is almost identical to the parent plant. So this method was vegetative propagation, in which the vegetative parts of the plant like the stem, roots and leaves were used for reproduction. Spore formation. This method of reproduction is used by the bread mold. Here is a picture of the bread mold, which we are also familiar. When bread becomes stale, it then develops this patchy black and white areas, and this is because of the formation of bread mold. Now the spore formation in Rhizopus, which is a bread mold, is made up of these structures. Here you have these long thread-like structures and they are called the hyphae. So this is the hyphae. At the end of the hyphae is a bulb-like structure called the sporangium. The sporangium contains the spores. Once the sporangium becomes mature, the sporangium then opens up. Here is the hyphae, and then we have the sporangium. The sporangium then opens up and releases these spores all over. In fact, if you notice in this bread mold, you find two kinds of areas. You have this white area here, and this is immature sporangia. The sporangia are still not mature. But you can, here you see there is this black mold. This black mold is when the sporangia are already mature and they release the spores. So this is from the textbook now. Even in many simple multicellular organisms, specific reproductive parts can be identified. The thread-like structures that develop on the bread above are hyphae of the bread mold. So the hyphae are these thread-like structures. They are the non-reproductive parts. They do not produce the spores. On the other hand, the tiny blob-like structures that are involved in reproduction, the blobs are called the sporangia, which contain cells or spores that eventually develop into new Rhizopus individuals. The spores are covered by a thick wall that protect them until they come into contact with another moist surface and can begin to grow. Let's go to the advantages of spore formation. First, a large number of spores are formed. The next one is the offsprings are similar to the parents because it is a kind of asexual reproduction. So the offsprings are very similar to the parents. And the third is these spores can withstand unfavorable conditions.